Yeah. Aha, fine, sorry. Now I see that there was a button mute on my microphone. So now I think that you can hear me. And I can start once more that there is endocrine system today, and that you can see in the first slide works now. Yes, fine. Thank you, Harsimran. Fine. So you can see a list of endocrine glands. This is a list of quite big glands. For example, thyroid gland belongs to. Sorry, it was mistake. Thyroid gland. What? Why does it working like this? Thyroid gland is one of big glands. Adrenal glands are also quite big. Pituitary gland also belongs to organs which are macroscopically well observable. But there are also some very small organs like pineal gland or parathyroid glands, and the endocrine system is special by the fact that there belong also microorgans like islets of Langerhans, which are distributed in the tissue of pancreas, or there are many isolated cells in heart, kidneys, gonads, placenta, or you already know DNAS cells which are found mostly in the digestive and respiratory tract epithelium. Yeah, so endocrine system is a quite special system which isn't based only on some organs connected by a tube like we know from digestive tract. Endocrine glands have one common feature. They are secreting hormones, and this hormonal secretion, hormonal signaling is very important knowledge of all medical students. So just introduction now, because you will hear about hormonal signaling in many other disciplines, in physiology, in biology, and then in most clinical subjects, pediatrics, internal medicine, and so on. So really very briefly today, there exist two. It's interesting. Why doesn't it write? No. It will be probably without drawing today. So how to something for the introduction. There exists, as you can see in this first slide or second slide, there exist hormones based on the proteins or peptides, sometimes biogenic amines. Usually these molecules are quite big. They don't pass through the membrane due to their size and also due to their hydrophilic properties. And so these protein, peptide or amine molecules have to bind to the surface of a target cell. There is a target cell, which should be influenced by our hormone, and hormone has to bind to its receptor. So the cell has got special integral membrane proteins or protein complexes in its plasma membrane, and hormone can bind to it. The conformation of this receptor protein is changing, and this change is is initializing a whole cascade of events in the cell, so-called second messenger answer, which goes to nucleus of a cell, which goes to mitochondria of a cell, for example, which goes to other structures of a cell, and changes physiology of the cell, and this is the effect of this our hormone. The second possibility are lipophilic molecules, usually small molecules, and the best example of such lipophilic hormone is steroid hormone. There are several steroid hormones. You already know some of them from the genital or reproductive system. Testosterone, for example, is steroid hormone, or estrogens, progesterone. And lipophilic or hydrophobic is also a hormone of the thyroid gland, thyroxine and trialthyronine. All these hormones, steroids and hormones of the thyroid gland can pass through the membrane. They are carried by some carrier proteins through bloodstream. Then near the cell they can leave their carrier, pass through the membrane by simple diffusion, passive transport through the bimolecular layer of lipids, phospholipids. They are now these molecules are now in the cytoplasm, and they can bind to the receptor, which can be usually in a nucleus. Yeah, so they don't need superficial receptors like hormones, which don't pass through the membrane. This bond of steroid receptor and hormone is or gives a result of the messenger RNA formation. So transcription is influenced by this bond, and the physiology of cell can change according to the type of the hormone. So these are two main types of hormonal signaling, and we will find among our glands today some glands which can secrete peptidic hormones like pituitary gland, pineal gland, some glands which secrete steroid hormones, mostly cortex of adrenal glands, and there are also glands which will secrete biogenic amines like medulla of adrenal gland, or already mentioned thyroid gland. The first of our glands is a hypophysis or pituitary gland. So let's look at it, and it will be also the first microscopic slide. Wait a little moment, I'll try to do something with my pen. It doesn't. Close window, and I'll try it once more. Just a moment. A big pen. Yes, and now it's drawing. So sorry, but now it works. So once more, a picture of pituitary gland, and you probably know pituitary gland from anatomy. You know that it has its anterior and posterior lobe. You know that it is connected with the diencephalon, particularly with the hypothalamus, by the stalk, which is called infundibulum. In histology, we are distinguishing two main parts of a pituitary gland, and these two parts are adenohypophysis, and the second part, which is also developmentally different, is neurohypophysis. Adenohypophysis doesn't form only the anterior lobe, but it is again another color. Adenohypophysis doesn't form only anterior lobe, but also something like a collar collar around the infundibulum, around the stalk, and the last part of adenohypophysis is a narrow area between anterior and posterior lobe. That means there are three parts of adenohypophysis. Pars distalis, it is the biggest part, forming almost the whole anterior lobe. Then pars tuberalis, which is a small part forming a collar around the infundibulum, and the last part of the adenohypophysis is pars intermedia, situated at the border between pars distalis and posterior lobe. Neurohypophysis has got two parts, which are histologically different. A big part forming a posterior lobe is called pars nervosa, and a smaller portion of neurohypophysis connecting the whole hypophysis and hypothalamus is the infundibulum. So there are 5 parts of the hypophysis. 3 of them belong to adenohypophysis, 2 of them belong to neurohypophysis. I think that we can look at the development of the hypophysis at this moment, because it can explain this double structure, or double histology. There is another presentation about the development of nervous and endocrine system. You can find it also on Moodle. By the way, these presentations are updated, so I recommend to download them once more. Mainly this histological one is updated. And if we look at the development of pituitary gland, it will elucidate this composition of the pituitary gland. Pituitary gland is developing from 2 sources. A yellow color was used for adenohypophysis, so I use it once more. Oh, sorry, I must click to this. Yellow color is used for this structure called Rathke's pouch. Rathke's pouch is the invagination of the epithelium of primitive oral cavity. Oral cavity, which during the embryonic development is called also stomodeum. You already know it because there was a development of face around it. So stomodeum is lined by ectoderm, of course, because it is the invagination of the surface of the embryo, and this ectoderm of the stomodeum forms a small pocket or pouch which is called Rathke's pouch. It is the structure giving rise to whole adenohypophysis. On the other side is a embryonic brain, which you don't know so much because the development of brain will be next week. Nevertheless, you know that there are some parts of a brain, and that there exists also a part of a brain called diencephalon. diencephalon. It is, in embryology, it is the second secondary brain vesicle. And this diencephalon has got a small outgrowth, small structure, growing against the Radke's pouch. This small structure, small process on the ventral surface of diencephalon is called infundibulum. And it will develop not only into the infundibulum of the definitive pituitary gland, but also into the pars nervosa. Second picture is showing elongation of the infundibulum and its dilatation into the pars nervosa. Its connection with the lumen of the encephalon is still preserved, and on the other side, Radke's pouch loses its connection with the oral ectoderm, remains situated in the head, in the skull. Bone is forming between the remaining part of Radke's pouch and surface of the mouth, of the oral cavity, and this separated tissue of the Radke's pouch is developing into the anterior lobe and into the pars intermedia and also into the pars tuberalis. Radke's pouch was hollow at the beginning. It was cavity in the center. This cavity is preserved even in the mature pituitary gland. We can find one or more cyst-like cavities in the adenohypophysis, and these cavities are found at the place where is a border of anterior lobe or pars distalis and pars intermedia. And we will look for them in our slides, and we will find them. They are lined by epithelium, which is developing from the oral ectoderm. So that's the development of the pituitary gland, which can also explain its double histology, let's say. So I go back to the histological presentation, and I can say something about the before I will start with the slide. I can say something about the general building schema of all endocrine glands. All endocrine glands, regardless of origin or function or location, have got two parts of their tissue. There is a stroma and parenchyma. Stroma and parenchyma. Stroma is very simple because it's every time reticular connective tissue. Reticular connective tissue is composed of reticular cells, which are very similar to fibroblasts, but have a little bit limited spectrum of products. They can produce only type 3 collagen, and that's why fibers of this connective tissue are only reticular fibers, because you already know that these fibers are composed of type 3 collagen. So reticular connective tissue forming the stroma of all endocrine organs is composed of reticular cells and reticular fibers. Point. Parenchyma. Parenchyma can be more variable. I use another color for this. Parenchyma can be trabecular epithelium, planar epithelium. It's exceptional, and this planar epithelium is arranged in follicles, And there are also two glands which haven't got epithelium as a parenchyma, but specialized nerve tissue. So there are three possibilities what can be the main tissue of the parenchyma. As you can see, trabecular epithelium is found in more glands, and also adenohypophysis, which will be our first gland, has got trabecular epithelium as the main tissue. Also parathyroid glands, adrenal glands, Langerhans islets. Neurohypophysis, which will be also demonstrated in the first slide, is a special nerve tissue. It's logical because it is a derivative of diencephalon, which is a part of brain, so it isn't surprising. Okay, so now let's really go to slide. The first slide, the slide of adenohypo or of the whole hypophysis, has a number 73 if you want to know it, and here it is in small magnification. If you want to ask something, don't hesitate to do it. I see your answers, I see your questions in the neighboring computer. There is a chat. So far there are also hello and works now, and we can't hear you, so go on, don't hesitate to ask if you don't understand anything or if you want to add something. So the picture here is a small magnification, the smallest magnification of the pituitary gland. So let's look at it. It's the section through the whole gland almost. We can see something like wings on both sides. These two wings are parts of pars distalis of the adenohypophysis. This central island here, this is a part this is a neurohypophysis. So here we can see neurohypophysis, pars nervosa. Here is pars distalis of adenohypophysis, and here is also pars distalis of the adenohypophysis. Here are some cavities. You can see smaller and larger cysts. They are remnants of the original cavity which was in the Radke's pouch, and so it's logical that this area has to be pars intermedia. For your orientation, I'll show you what is the direction of our section. I'll go to presentation for a moment. So I think that this our section is something like, oh no, sorry, is something like this. Yeah, so we can see a small part of neurohypophysis, and in both sides, in one here and one on the other side, is a pars distalis. No, it's here written. Yeah, okay. So we can see 2 times pars distalis, 1 central pars intermedia, and a circular section through the pars nervosa. let's go back to slide. And let's look at the tissue at first of pars distalis in a little bit higher magnification. In this small magnification, we can see just very rough arrangement of cells here. Nevertheless, we can see something. We can see cords, anastomosing cords of cells. We don't know anything about the type of cells in this magnification, but we can see that they are very tightly arranged. So we can suppose that it can be epithelium. Let's look at it in a high magnification now. Yes. So now we can see more. We can see such clusters of cells. Oh, sorry. Clusters of cells, and arrangement of these cells is very tight. So really is a proof that it is epithelium. It is typical example of trabecular epithelium. You know already liver as the example of trabecular epithelium. There it is little bit, its arrangement is little bit different. There are usually cords or trabecules arranged of only two cells. Here in the adenohypophysis, the thickness of trabecules can be bigger, so there are more cells arranged in the trabecule. These cells are tightly associated, but they have basal surfaces oriented towards vessels. There are many vessels situated in septa, going through the I use another color for vessels. So here is one vessel, here is another vessel, here I see a vessel, here is a vessel. These vessels are quite wide, quite dilated, but have a very, very, very thin wall, and we know that these vessels situated among the cords of epithelial cells here are sinusoids. They are special atypical capillaries, sinusoids, because we are in pars distalis of adenohypophysis. So the arrangement of the adenohypophysis are cords of epithelium, vessels, and of course some connective tissue which is accompanying vessels and which is separating cords, and it is this stroma. It is reticular connective tissue of the stroma. Epithelial cords of pars distalis are composed of several types of epithelial cells. I'll use even higher magnification, and we will try to recognize cell types in the epithelial cords. So let's look at any randomly chosen cord. We can see that some epithelial cells are red. You all are experienced histologists, so you know that the red color in synoptic histological staining means eosinophilia or acidophilia, simpler. These cells, which are red in hematoxin and eosin staining, are called acidophilic cells. So the first cells of the pituitary gland parenchyma are acidophilic cells, or simply acidophils. Second cells, which aren't so prominent, but we'll find them as well, have a cytoplasm light purple. Its color is similar as the color of a nucleus, and it is a proof for you histologists at the end of the histological course only that the cytoplasm of these cells is basophilic. Everything what has the same color as a nucleus is basophilic, and so there are also basophilic cells or basophils, but be careful, basophils are also granulocytes, basophilic granulocytes, so here are different basophils. Let's try to find another basophil. So here was one, here is a basophil, some typical basophilic staining is also here and here, and also here is a basophil. Here it is quite dark basophilia of the cytoplasm. So these are basophils. We can find also cells which don't stain by any of these two basic colors. They don't stain by eosine or by hematoxylin. They are so-called chromophobic cells. Chromophobic is, for example, this cell. Chromophobic is this cell. I don't see. Oh, maybe it's slightly eosinophilic. But what about this cell? It's white and has only a nucleus. Maybe also this cell or this cell. So in fact, to summarize it, we can find three staining types of epithelial cells in trabecular epithelium of adenohypophysis: chromophobic, acidophilic, and basophilic. These acidophilic and basophilic cells can be together taken under the term chromophilic. So there are chromophilic cells, which can be either acidophilic or basophilic. Basophilic, and there are also the second category, or maybe third, are chromophobic. They have phobia from colors. Chromophobic, chromophilic. Yeah? Chromophilic like color, chromophobic hate color. I see some question. What causes basophilia in the cytoplasm of these cells? Yes, I explain it. I explain it why some cells are acidophilic, why some cells are basophilic, and why some cells don't stain well. At first, chromophilic cells are active cells. Chromophilic, that means acido and basophilic cells, are active cells which are synthesizing and secreting hormones. Adenohypophysis can produce at least 6 types of hormones. So it was discovered that there are 5 or maybe 6 types of secretory cells. But we can see only acido, basophilic, and chromophobic. So how to explain it? It was found that, and maybe I can use now a schema which can help us. there was discovered that acidophilic cells are in fact cells of two types, somatotropic and, it isn't here written so I will write it, somatotropic cells they belong to acidophilic, so somatotropic, and mammotropic. Somatotropic cells secrete growth hormone, which is also called somatotropic hormone. So you can find both these names and both these abbreviations, STH or GH, growth hormone or somatotropic hormone, which is very important. If you look at the schema, you can see that this growth hormone is very important for bone growth, for muscle metabolism, but for adipose tissue metabolism. Altogether, it's very important mostly for the development and growth. So mostly important during childhood, during this period when we are growing. Mammotropic cells secrete prolactin. It is here written. Prolactin, which is also called luteotropic hormone, so that's why there is this second abbreviation. And this prolactin has one main target organ, and it is a mammary gland, and it is very important for milk secretion. So very important is mostly for pregnant and breastfeeding women. So there was discovered that there are two types of acidophilic cells, but we don't distinguish them at the level of light microscopy. We can try to distinguish them in the level of the electron microscopy, or we can distinguish them by immunohistochemical proof of hormones. And both these hormones, prolactin and growth hormone, are glycoproteins, which have basic pH, and that's why secretory granules and thus also the whole cytoplasm of this cell is eosinophilic, acidophilic. Basophilic cells are of three types, maybe four, it isn't still clear. The first type of basophilic cell is thyreotropic cell. Thyreotropic. It is secreting TSH, thyreotropic hormone or thyroid stimulating hormone. It is a hormone which is stimulating thyroid gland. We will speak about it. The second are gonadotropic cells. Gonadotropic cells are maybe special by the production of two hormones, FSH and LH. You know them mostly in the female reproductive system. We spoke about it. FSH is follicular stimulating hormone. It stimulates the growth of ovarial follicles. Luteinizing hormone LH is a hormone which stimulates transformation of ovarian follicles into the yellow body, corpus luteum, and then to the production of progesterone. They are also active in males, so FSH is important mostly for spermatogenesis, and so its target cells are mostly Sertoli cells. They produce then androgen binding protein, and luteinizing hormone is important for the testosterone secretion because they stimulating Leydig interstitial cells. So there are two types of gonadotropic hormones. Some theories say that there is a cell which can secrete both of them. Another theory is that there exist two types of gonadotropic cells, G1 and G2, and one of them can secrete FSH and the second can secrete only LH. It isn't completely discovered so far. Next type of the basophilic cell is corticotropic cell. Corticotropic cell can secrete several hormones, but the main of them is adrenocorticotropic hormone, ACTH, which is stimulating cortex of, here it is, cortex of the adrenal gland, adrenal cortex. But ACTH is secreted as a precursor, and some of the derivatives of this precursor is also melanocyte stimulating hormone or beta endorphins, some lipotropic hormones. So there are more hormones which are developing or which are forming by digestion, lysosomal digestion of the precursor of ACTH. So at least 3 types of basophilic cells and 2 types of acidophilic cells. And I still didn't say why these cells are basophilic. They are basophilic because the products of these cells are pure peptides which are not glycosylated, and so mostly the color of ribosomes or the staining property of ribosomes is a reason of their basophilia. And there are also chromophobic cells, and these chromophobic cells can be either not fully developed chromophils. Chromophils are differentiating from some stem cells, and during this differentiation they haven't got so many granules to be stained by hematoxin or eosine. So it is one possibility of the chromophobe formation. Second possibility can be very old basophilic or acidophilic cells. They lose granules, they finish their secretory cycle, they are preparing for apoptosis, and so they are also chromophobic in this period of their life. And the third theoretical possibility, what can be chromophobe in this trabecular epithelium, are cells which are called follicular stellate cells. I'll find them in the presentation. There is one electron micrograph. Oh no. Electron micrograph is here. Yeah, you can read follicular. Oh, it's black on black. Follicular stellate cell. Follicular stellate cell is, for example, this one. It is a cell with a small nucleus, and its cytoplasm has got processes. Here is the second one. And there aren't many things known about them, but probably they are supporting cells of this secretory. In this case, it is gonadotropic cell, for example. So chromophobic cells can be either not fully developed or too old chromophilic cells, or folliculostellate supporting cells. Let's go back to our slide. Sorry. So we can see once more chromophils, which can be basophilic or acidophilic or basophilic, and chromophobes. Among them, reticular connective tissue, sinusoids, it was already demonstrated. I think that pars distalis can be finished at this moment. Let's go back to small magnification. And let's go to pars intermedia. Pars intermedia is at the border between pars distalis. Pars distalis is here. Pars nervosa, it's here. I will describe it in a moment. So pars intermedia is here. This is pars intermedia in the middle magnification. This structure, here, or also this big one, is a remnant of the original space in the Radke's pouch, and we call these structures now Radke's cysts. Radke's cysts. It's bad. Radke's cysts. They are very variable. Somebody has got one big cyst, somebody has got, as in this case, several small cysts. Sometimes we don't find any, but usually yes. When we look at the tissue around these cysts, it's pars intermedia. Pars intermedia is composed mostly of cords of basophilic cells. Eosinophilic or acidophilic cells are not many here. So if you look at, for example, this cord here, you can see basophilic cells. I think that this color of them is well recognizable, prominent. Here is another group of basophilic cells, and very exceptionally there can be some eosinophilic cells, like in this case or in this case. Of course, there are also chromophobes. Chromophobes have to be everywhere. So chromophobe is, for example, this. Chromophobe can be this, or in this cord here is nice chromophobe, or here is a chromophobe. Again, capillaries, sinusoids, but these sinusoids become smaller and gradually change into fenestrated capillaries. And here is a small connective tissue septum, and in this direction we will go to neurohypophysis. So let's look at it. Neurohypophysis has a completely different structure than adenohypophysis. It is specialized nervous tissue. It is a part of diencephalon, in fact, and it is a part of its white matter only. You already know something about white and gray matter of CNS. Gray matter has got bodies of neurons and also many other structures, and white matter hasn't got bodies of neurons. It has got only processes of neurons, axons of neurons, and it is the case of neurohypophysis. It is composed of many axons of neurons which haven't got bodies in the hypophysis but in the hypothalamus. You probably know pictures, schemas, but we can look at some of them once more. For example, here is the schema. So bodies of a neurons having their axons in the pars nervosa are situated in hypothalamus, particularly in two big nuclei, nucleus paraventricularis and nucleus suprapopticus. There are also smaller nuclei in so-called eminentia mediana. The biggest of them is nucleus arcuatus. So all these red nuclei here are important for us now because they contain bodies of neurons which send their axons into the neurohypophysis. Some of them, mostly these from nucleus arcuatus, can reach only infundibulum, and the axons from nucleus paraventricularis and supraopticus are longer and they go to pars nervosa. So when we'll go back to our slide, we can see many structures which are branching here, and these are mostly unmyelinated axons of neurons from hypothalamus. We can see some cells here. We can see nuclei mostly. For example, here is a nucleus and small cytoplasm. Here another one. Here as well. Here as well. We already know that the nerve tissue isn't composed of neurons only, but that there are also so-called glial cells, which are mostly supporting. Glial cells of neurohypophysis are very similar to astrocytes, but they have another name. They are called pituocytes. Pituocytes and axons form the main structures of neurohypophysis. There are also vessels, of course. For example, here the bigger vessels are situated in septa. Smaller vessels, capillaries, which are fenestrated capillaries, are situated directly in the nerve tissue. Where is some capillary? No, here is, for example, one erythrocyte situated directly between axons. It's almost capillary, little bit bigger than capillary. And here, for example, is a very nice capillary. We can see only one erythrocyte and endothelial cell nucleus, and you can see that this is situated directly among the axons. So there are many capillaries and axons, and that is very important because products of neurons which are secreted in the bodies, in the nucleus paraventricularis and nucleus supraopticus, are transported by axonal transport to the dilated buttons at the end of axons, which are similar to synaptic endings, but the synapse here isn't the synapse with any second neuron or any target cell, but these synaptic like endings are sitting on the surface of these fenestrated capillaries. So hormones which are released from the cells after the impulse are secreted directly to bloodstream, and it is important for all endocrine glands. They haven't got any ducts, and they have to contain a lot of capillaries which are receiving hormones from secretory cells, in this case from neurosecretory neurons. So there are pituocytes, axons, capillaries, and of course some reticular connective tissue forming again the stroma and surrounding bigger vessels. We can find special structure in the neurohypophysis. I will show it in the schema. Neurohypophysis, yes, and here is schema of the axonal ending, as I described it before a moment. It is axonal ending on the surface of the capillary. So here is the area where hormones are secreted and where they are going directly to the capillaries. These hormones are stored in secretory granules, and these granules can accumulate in some dilated parts of axons during their transport, and these accumulations of secretory granules are called herring bodies, and these herring bodies are sometimes big enough to be visible in a light microscope, so I'll try to find them. So let's go back to the slide. And in this high magnification, when I look at the neurohypophysis, I can sometimes find granular structures. Wait a moment. Herring's body. I'm sure that there are some bodies. Just to find them. Yeah, here is one. I don't know if you believe me, but this structure here is a herring body. It isn't any cell, but only dilated part of an axon containing many granules, and these granules are visible in a microscope. Sometimes you can find also pigmentations here in this area of neurohypophysis. Some axons can be pigmented by neuromelanin. It is possible. Which hormones are stored in these herring bodies and then secreted to capillaries of the neurohypophysis? These are two hormones, probably you know them. Mostly one of them was already mentioned during the urinary system. It is ADH. So the first hormone is ADH. It is the abbreviation of its name, antidiuretic hormone. It's also called vasopressin. It's influencing collecting tubules of kidneys, and it's also influencing smooth muscle vessels to contract them and to increase the blood pressure. And the second hormone, which is mostly important for females, is oxytocin. It is very potent hormone stimulating smooth muscle cells of the uterus during parturition, and it also stimulates smooth muscle cells and myoepithelial cells of mammary gland during breastfeeding. So there are two types of hormones. Each of them is secreted by another neuron, so this is the same as in the adenohypophysis. One cell can secrete only one type of hormone, ADH or oxytocin. The nuclei secreting them, or nuclei where are bodies of these neurons, are nucleus supraventricularis and nucleus paraventricularis and nucleus supraopticus. And it was discovered that paraventricular nucleus secretes primarily oxytocin, and supraoptic nucleus secretes primarily antidiuretic hormone, but both nuclei can contain neurons secreting both these hormones, so it isn't, so it is here written primarily, yeah? So I think that we can slowly finish a slide and the problems of hypophysis, of pituitary gland. Maybe, yes, maybe last thing, let's look at electron micrographs showing secretory cells of adenohypophysis. Secretory cells of adenohypophysis, that means these basophils or acidophils are all of the same histological type. They all belong to polypeptide secreting cells. But if you look at them in the electron microscopy, you can see that they can be variable. You can see, for example, this cell, which is labeled by a letter S, which it is not good, this blue color. I should use red color because it is one of acidophilic cells. It is somatotropic cell. Here is another one. These are cells having quite big and very many secretory granules. Neighboring cell, which has the label C, belongs to basophilic cell. It is corticotropic cell, and you can see that it has only few very small granules. So there is a possibility how to recognize them not only in light microscopy but also in the electron microscopy. This cell up, it has a label T. It isn't very well visible under these here descriptions, but it is T thyreotropic gland, and you can see that it has many small granules. The size of these granules is similar like here, but they are much more numerous. Here are also gonadotropic cells. Unfortunately, we don't see any mammotropic cell in this picture. And we can see sinusoid also. We can see sinusoid having fenestrations. Then this sinusoid can have also pores. You can see that it's quite big vessel, and granules or content of granules, hormones situated here in these granules can be secreted from a cell, and they go directly to bloodstream through these fenestrations or pores in the wall of the sinusoid. Okay, so I think that we can really finish the hypophysis, and we can go to next gland. Next gland will be one of very small glands, which is also situated in the area of diencephalon, and it is so-called pineal gland or epiphysis. Pineal gland pineal gland or epiphysis is really a very small gland. It is the picture from gross anatomical dissection, and you can find this gland here. I don't know why it's so thick. You can find this gland here between mesencephalon. You are recognizing hopefully corpora quadrigemina superior colliculi, inferior colliculi. So between mesencephalon and the roof, this is a roof of the third ventricle here. Ventricle. So it is situated ventricle. It is situated in the area of epithalamus, a part of diencephalon. Pineal gland is histologically much simpler than pituitary gland, and so let's look at its slide. Its slide has a number 74, and we can see it now under the microscope. It's almost whole gland, so we can see some connective tissue on the surface. It is in fact pia mater, it belongs to meninges, and in this case connective tissue from the pia mater is going in and is dividing the tissue of our gland into many lobules. So there are septa, you can see septa of connective tissue which are anastomosing, and they are dividing our organ into lobules. So it's one lobule, second lobule, third lobule, this lobule is a little bigger, and so on. Also, the typical feature of a pineal gland is like lobulation, which you know from exocrine glands mostly. Nevertheless, this gland belongs to endocrine glands because there isn't any duct, of course, but its products are going to blood again. Pineal gland, together with neurohypophysis, are special endocrine glands having nervous tissue inside. So let's look at the tissue of the gland now. I'll use higher magnification. You probably remember the structure of neurohypophysis, which was before a while, and you can see that it is very, very different. Nevertheless, it is nerve tissue, but it is another type of nerve tissue. Neurohypophysis was specialized white matter of CNS, and this pineal gland is specialized gray matter. That means it can contain neuronal bodies. And really, most cells found here are neurons. How to recognize it? When I use high magnification, we can really see many cells having typical neuronal nuclei. You hopefully know that each neuron has got spherical, very light nucleus with prominent nucleolus in the center. So, typical nucleus of a neuron, another typical nucleus of a neuron, another typical nucleus of a neuron, and there are very many. Most cells here in this area has got this typical nuclei, and these cells, these special neurosecretory neurons of pineal glands, are called quite simply pinealocytes. So there are pinealocytes, which are neurons. They have quite short axons, which are ending on the surface of neighboring vessels. For example, here is some vessel, but it is a vessel in the septum, but another erythrocytes directly in the area of this pinealocyte can be found. I'm sure that there must be some vessel, or vessel can be also empty. It isn't necessary to see erythrocytes inside. So there is a vessel, here is a vessel, and so the short processes of these our pinealocytes are ending on the surface of these small fenestrated capillaries. So here are again fenestrated capillaries, and secrete their hormones. Hormones of or the main hormone of pinealocytes is called melatonin. Melatonin is a biogenic amine, quite simple and small molecule, which is very important for circadian cycles, for sleeping, also for sexual cycles, for the maturation of the CNS and also maturation of gonads. So it has a lot of important functions. The pinealocytes can secrete not only melatonin, but they can secrete also another amines like serotonin also was here found, or another simple molecules, but melatonin is the main hormone of pineal gland. I see a question from Hasim Ranvirk. Is the gray matter still from the encephalon? Yes. Epithalamus and hypothalamus both are parts of the encephalon. Epithalamus gives rise to neurons of pineal gland, so to these pinealocytes. Hypothalamus contains neurons which send axons to neurohypophysis. So yes, it's everything from the diencephalon. Each nerve tissue has to contain glial cells. So there are also glial cells. So not only pinealocytes are here, but we can see many smaller cells with smaller, often ovoid nuclei. Most of them are astrocytes, but there are also especially small and dark nuclei of microglial cells, which you already know, and you will try to recognize them next week in many organs of CNS. So astrocytes, microglial cells, maybe some oligodendrocytes can be here as well, because some myelinated axons of the visual tract are going to the pineal gland, because pineal gland is dependent, or the function of the pineal gland is dependent on light. It works and secretes more melatonin at night. That means when dark is outside, and it is inhibited by light. So it is connected with visual tract. Okay, so we can see trabecule-like arrangement of pinealocytes, some glial cells, septa, vessels, and so those are the main structures of the pineal gland. You already saw it in small magnification. I go back to small magnification now. So just focus. And probably you mentioned that there are some structures which I didn't describe. Here is one. Here is another one. This. Some of them are so big that the tissue around is destroyed because they were hard and they were interfering with the knife during sectioning. These structures are called corpora arenacea. But maybe you know them better under the name of English name brain scent. Brain scent, in Latin acervulus cerebri. And it's in fact a good name. It is really scent. There are calcified structures originally accumulation of some proteins, proteins of melatonin binding, or there are melatonin binding proteins which are secreted together with melatonin and which accumulated in this area. So the amount of this brain scent is increasing with age. Children haven't got it, and old people have got a lot of big grains of brain scent in the pineal gland. You can find similar brain scent also in choroid plexus and in some other locations of pia mater. You can see that they are mostly situated near the surface, but some of them are present in the center. Let's look at some particle of this brain scent in high magnification. Oh, I lost it. You can see here, for example, quite well that it has a lamellar structure. You can see several lamellae, which are proofs of gradual growth of this structure. So it was at the beginning there was just small grain, and it was growing during the life, and the proof of this long growth is the lamellar structure. Here it was visible, but I'll find another one. Oh, this is destroyed because it was too hard. Here is this small grain of brain scent has got very nice lamellar appearance. How do you see it? These we don't know any function of this brain scent. Probably there are really only remnants of proteins which were dystrophically calcified, but they are very good for medicine, because the pineal body of old people is X-ray contrast. We can see pineal body as a dense structure in the picture, in X-ray pictures or in computer tomography. And as we know that this pineal body is in the roof of the third ventricle, we can recognize the position of the third ventricle of a brain, which can be very important in some situations, in injuries or in the tumors, where the neurologists want to exactly know the position of the third ventricle. Okay, so I think it can be enough about the pineal body or pineal gland, and I think that we can have a short break now. It's in my computer written here 2:19, so let's meet once more at 2:30. That means in 11 minutes, in 2:30 of the Czech time, of the time here in Prague. So see you in a moment. Yes, so I'm back. I hope that you'll be back in a moment as well. And during the break, I read a question of Jean-Pierre Bischoff about the acervulus cerebri, but there is written that I just answered this question. In fact, yes, acervulus cerebri hasn't any known function, so it probably doesn't affect the function of pineal gland in old persons. Before I'll start to speak about next gland, which will be adrenal gland, I will go back for a moment to hypophysis. I forgot to say anything about blood circulation in hypophysis, and it was a mistake, because blood circulation of hypophysis is very important and very interesting, as you can see in this schema, and maybe as you know also from anatomy. The hypophysis has got two arteries, superior hypophyseal artery and inferior hypophyseal artery. Superior hypophyseal artery is going from internal carotid artery to the area of infundibulum and pars tuberalis of adenohypophysis. In this area, it branches into many capillaries, and so-called primary capillary plexus is here formed. Primary capillary plexus is formed in the area of infundibulum and pars tuberalis. Capillaries of this primary plexus are fenestrated. These then join to several hypophyseal portal veins. There are hypophyseal portal veins. They go to pars distalis and branch once more into the network of secondary plexus, secondary capillary plexus. And this secondary capillary plexus is composed of sinusoids. So there are fenestrated capillaries in primary plexus and sinusoids and in the secondary plexus. And these sinusoids anastomose back to veins, to hypophyseal veins, which go to sinus cavernosus. You can see that inferior hypophyseal artery is supplying only neurohypophysis. So neurohypophysis has got independent blood supply. The fact that there are two capillary plexuses connected by veins can say us that there is so-called hypophyseal portal system. It is a venous portal system. That means that there is a vein between two capillary networks, two capillary plexuses. Why there is this organization? This is because nucleus arcuatus and other nuclei in hypothalamus, in this area which is called median eminence, secrete hormones which are going only to the infundibulum. They have short axons, and these hormones are liberated here or secreted here to the primary capillary plexus. These hormones from these nuclei in the hypothalamus are so-called hypophysiotropic hormones. Hypophysiotropic can be releasing or inhibiting hormones. Something about them is here in this schema. Some of these hormones are stimulating, for example, growth hormone, releasing hormone is stimulating somatotropic cells. On the other hand, somatostatin is inhibiting somatotropic cells. The same somatostatin is inhibiting also the thyreotropic cells. This topic is quite complicated, and there isn't place in the first year histology isn't place to explain everything. So just know that there exists some neurosecretory neurons in the hypothalamus which secrete hypophysiotropic hormones. They are secreted here in the area of the infundibulum, and they are transported by hypophyseal veins into pars distalis, where they directly influence, so there is transport by several hypophyseal veins from one plexus to second plexus, and they directly influence acidophilic or basophilic cells in the adenohypophysis. So that's the principle of this special circulation in the hypophysis, which I forgot to describe before the break. And now we can go on, and we can go to adrenal gland. So I'll find it in our presentation. It is near the end of the presentation. Parathyroid, adrenal or suprarenal glands. These are two glands situated on the cranial poles of kidneys. Something about their anatomy is in this slide. You can find it in the presentation. They have their own capsule, but they are incorporated in a big adipose capsule together with kidneys. And let's look at their histology. I have already this slide in the microscope. It has a number 75, and we can see an adrenal gland. We can see its natural surface here in this area, and this area it is sectioned, so it is bigger than was possible to place on glass, so it's quite big gland. On the surface we can see a capsule. This capsule is composed of a thin layer of quite dense connective tissue. You can see it here. Several layers of collagen fibers and some fibroblasts. So this is capsule of a gland. It doesn't send some big septa going to the interior of a gland, so this gland isn't divided into any lobules. Just very thin bundles of reticular fibers are going into the gland from the capsule, and they are bringing vessels into the interior of a gland. Around this fibrous capsule there is adipose capsule. There was some place where here, for example, is a remnant of this capsule. That means fat. No, that means some fat. You can see adipocytes here, and it is a capsula adiposa renis, which is surrounding both adrenals and kidneys, and it is very well vascularized. You can see capillaries here, and here is also veins, so bigger vessels are here. We can look at the vascularization of a kidney, but at first let's finish this brief description. Just under the capsule there is a cortex. Kidney has got cortex, and kidney also, but adrenal has a cortex and inside, but it's problem to find it in this big magnification, but believe me that inside will be a medulla. So now let's look at the cortex at first. You can see that the cortex is composed of cords. These cords have got very variable shapes. Here under the capsule there are arcuate cords, or they can resemble glomeruli, something like tufts of cells spiralized. The cells of these subcapsular cords are quite small and highly eosinophilic. You can see there almost red color. When you go deeper, you can see that the cells become bigger, lighter. They have got a lot of white spots, which are lipid droplets, and they are arranged in straight fascicles, straight cords. Yeah. So one part of the cortex is composed of small cells arranged in arcuate or glomerular cords. Another part is composed of fascicles of cells, and when we'll go, but I must at first go to smaller magnification to find some orientation. When we'll go to the deepest areas of our cortex, I remain in this middle magnification. Deepest areas of the cortex are here. We can see that the cells are still quite big, but they are again a little bit darker, more eosinophilic than were here. And they are arranged in irregular network. Cords here are arranged irregularly. All these sublayers of the cortex are composed of trabecular epithelium, but trabecules of this epithelium have got different shape, and so there are 3 zones of the cortex, which I'll show in the schema. Schema is here, and this is the schema which I am looking for. So the superficial layer of the cortex, where are really small epithelial cells with high level of eosinophilia, arranged in arcuate or glomerular cord, is called zona glomerulosa. Middle layer, the thickest part of the cortex, where the cells are much bigger, full of lipid droplets, just lighter than were in zona glomerulosa, and arranged in straight, parallelly organized cords, is called zona fasciculata. And the deepest layer of the cortex, where the cells are again smaller, more eosinophilic, with less lipid droplets, some pigment there can be, I will show it, and the arrangement of cords is in irregular network. It's called zona reticularis. So there are 3 zones of the cortex, and I will show them once more in the slide. So let's go back to the surface. Capsule was already described, and the zone of the cortex under the capsule in this depth approximately is zona glomerulosa. The cells of this zone are secreting mostly aldosterone and other mineralocorticoids, which are influencing salt metabolism, mainly reabsorption of sodium in distal tubules of kidneys. When we'll go deeper, we are in zona fasciculata. Cells here are bigger, have got more lipid droplets, and secrete glucocorticoids. So there were mineralocorticoids, and here in zona fasciculata are mostly glucocorticoids. Secreted. Zona fasciculata is really very thick, so I will go through this zone. They are still in zona fasciculata, and I am in a bad place because here is one small zona glomerulosa. It is some invagination of a surface, but here in this place is a border with zona reticularis. So I'll try to show it. This is still zona fasciculata, and here is zona reticularis. You can see irregular arrangement of little bit more eosinophilic cells. Compare the eosinophilia of this cell and this cell, for example. This much lighter, also bigger than this first one. So this is zona reticularis. Cells in this zone are gradually degenerating. You can see that they have darker nuclei. The nuclei can become pycnotic, like in this case. And also some pigment, lipofuscin, is often found in their cytoplasm, and their secretion is very variable. They still can secrete some glucocorticoids, but some of its hormones, some of the hormones secreted by these cells have activity of androgens. So androgens are probably the main product of zona reticularis. So zona fasciculata and zona reticularis. And when we'll go deeper, we can find another interesting border here. Oh no, it was not well drawn. I will be more precise. I can really very precisely draw a border between two tissues. This is still zona reticularis. That means a part of cortex, and here I use blue color is medulla. You know, hopefully, that the adrenal gland is composed of cortex and medulla. They are organized very similarly, but the origin and the secretion and cell type of both these parts is very different. And here in this picture you can see a difference in color. Cells here in the medulla are basophilic. They have a cytoplasm of the same shade, only lighter than as a nucleus. Cells here in cortex are eosinophilic. Different color of cytoplasm and nucleus. Yeah? So basophilic cells in medulla, eosinophilic cells in the cortex. I will go back to cortex and I will show an electron micrograph which will elucidate why these cells are eosinophilic, why these cells have got these white grains inside, and so what is their function and type of secretion. This electron micrograph is found in this presentation as well, I hope, or I must find it in another one. Wait a moment. Yes, this electron micrograph is almost this one. So no, I must go to another presentation. Wait a moment. This electron micrograph belongs to the examination electron micrographs, and it is an electron micrograph of one cell in the adrenal cortex. What do we see? We can see here a surface of our cell, which is quite complicated. The cell sends some processes like microvilli, so this is complicated surface of our cell. Here is the basal lamina of this cell, because it is epithelial cell, isn't it? And here, this structure in the corner is a capillary. And you can see that it has fenestration here, but it has a pore as well. Capillaries in zona fasciculata mainly are atypical or sinusoids. So there are sinusoids in the zona fasciculata and fenestrated capillaries in other zones of the cortex. Remaining part of the picture is a cytoplasm. Only here down is a very small piece of a nucleus, maybe. But all here is a cytoplasm, and what do we see in the cytoplasm? We can see three very typical structures which should help us to recognize the cell type, and the first and maybe most important of them is this structure. I think that you already recognized tubular mitochondrion. So there are tubular mitochondria in these cells. What does it say to experienced histologists like you? It says that these cells should secrete steroid hormones. Another helping features of the cytoplasm is a lipid droplet and a lot of small tubules and cisternae, but not in this section, of smooth endoplasmic reticulum. So lipid droplets, smooth endoplasmic reticulum, and tubular mitochondria is a typical triad of cytoplasm of steroid secreting cell. And where are many mitochondria? The cytoplasm is eosinophilic, it's already known. And where are lipid droplets? We can see white grains in the light microscopy after the extraction of the lipid content. So it completely corresponds with our light microscopic picture. All the cells of the cortex are secreting steroids, and all hormones of the cortex, that means mineralocorticoids, glucocorticoids, and androgens, are steroid hormones. In fact, the adrenal cortex is the biggest accumulation of steroid secreting cells at all. You already saw steroid secreting Leydig cells in testes, you saw thecal cells or granulosa lutein cells or thecalutein cells in the ovary. They were secreting steroids as well. So it was electron micrograph, and now we can go back to slide. Yeah, so all these cells here, which I now can show in high magnification, all these cells here forming cords, having network-like appearance in this area because it was zona reticularis, or fascicular appearance, that means parallel, straight direction in zona fasciculata, or here near the surface, irregular arrangement again, zona glomerulosa. So all the cells here are secreting steroids. Very thin septa among them, which are filled with reticular connective tissue, should contain capillaries. For example, here I see an erythrocyte, and these capillaries, mostly in the area or zona fasciculata can be sometimes dilated, like in this case. You can see more erythrocytes in one thin-walled vessel, and it is typical picture of sinusoid, or here is a sinusoid. You also remember that zona fasciculata has got sinusoids, and zona glomerulosa and zona reticularis has got just smaller capillaries where only one erythrocyte can be, or here there is a chain of erythrocytes, but in the cross section theoretically no more than one erythrocyte can be there. So it was a cortex. Maybe briefly blood supply. Sorry, it isn't good picture. Blood supply, no. The schema showing these zones here has got also blood supply, which is quite important. Arteries of the adrenal, each adrenal gland has got superior, middle and inferior artery. Each of them is going from another bigger artery, and they form capsular arteries which branch into so-called subcapsular plexus. The subcapsular plexus is supplying mainly zona glomerulosa, then is forming sinusoids, adrenocortical sinusoids going through the zona fasciculata, and then a network of capillaries in zona reticularis, but it isn't any portal system, because these middle vessels are not veins or arteries but sinusoids, which also belong to capillaries. And blood then goes to medulla. But medulla, if supplied only through these cortical capillaries, would suffer from lack of oxygen and so on. So that's why there are also medullary arterioles, which are also called penetrating arterioles, which are going through the whole cortex without any branching and participate in the formation of capillary network in the medulla, and all these capillaries then go to central adrenomedullary veins, which are situated in the medulla, and which then go to adrenal vein. And it was a small introduction to the medulla, so let's go back to slide, and in small magnification I will find the medulla once more. So medulla is here in the center of our specimen. So medulla is this area. No, it isn't so precisely surrounded by. I think that you can see a difference of color of these cells here and these cells here. This is the cortex, this is a cortex on the other side, and this is medulla. Cells here are eosinophilic, cells here are basophilic. But everywhere the cells are arranged in trabecular epithelium. There are two different trabecular epithelia in this gland. Let's go to medulla in high magnification. Where is it? Here it is. Yes. So once more a difference of medulla and cortex. Cortex is now at the right side, so here is a border. Here is cortex, zona reticularis, and here is medulla. Look at the different color. Cells here in medulla are a little bit bigger than were in the zona reticularis, but not then in zona fasciculata. Here is zona fasciculata. You can see the cells are here big. They are slightly basophilic. They surround quite big veins. This is a vein, Oh, here is some artifact, but let's say that here is the lumen of one vein. Maybe some better vein will be here. Yes, so they are medullary or central veins where all capillaries are going. Cords of a medulla are composed of cells secreting biogenic amines. They are called catecholamines, and they are quite well known. Two types of catecholamines there exist. That means two types of cells which are there secreting, and one of them is called adrenaline, or also in some another English textbook, epinephrine. Epinephrine is one product of medulla, and the second is noradrenaline or norepinephrine. And they are secreted in two different cell types. So two types of cells are in medulla, but they are not recognizable at the level of light microscopy. So we can see just basophilic cells, typical cells secreting biogenic amines. Some cells of medulla can be also neurons. There can be ganglionic neurons. I explain it in the part where will be a development. But I didn't find them in this slide, so I will not demonstrate ganglionic cells. Just cords of the catecholamine secreting cells. In between there are again capillaries, fenestrated capillaries, and of course reticular connective tissue as well is here, because each gland needs its stroma. So this is a medulla, and maybe I can show also electron micrograph of the medulla and then something about development. I think that the electron micrograph of the medulla will not be surprising. Oh. For example, here, or maybe better is here. Oh, it's very poor resolution of this picture. Nevertheless, there are small parts of two cells. One of them is secreting epinephrine. It is this. It's E here. Second, norepinephrine. It is here. Here is a border, and you can see that the granules of these two cells are a little bit different. The granules of norepinephrine secreting cells have got a darker central core than granules of the cells secreting epinephrine, but both these cells are from the group of biogenic amines or catecholamines secreting cells. So they are very similar. They bind silver salts and chromium salts, so that's how we can recognize them also in the light microscopy, and so they are also called chromophilic or chromaffin or argyrophilic cells, but it's just detail. And several words about the development of adrenal gland. It's very interesting. Adrenal gland has a little bit similar development as a pituitary gland, because it has also two sources, two tissues at the beginning. The maternal tissue of the adrenal cortex is celomic epithelium. Celomic epithelium, you already know. It is the epithelium lining the abdominal cavity. Celomic epithelium is differentiating into mesothelium, for example, lining all the cavity, but it's also differentiating into the genital cords or sex cords, which were very important during development of gonads. And very similar is the situation in the area of adrenals. When you'll go to next picture here in this presentation, there will be visible that adrenals or suprarenal glands and gonads are developing, in fact, in one place in urogenital ridge. So nothing surprising that the development of adrenal cortex and gonadal sex cords is very similar. So coelomic epithelium is growing and forming cords which are transforming into the cortex. Medulla has got very different origin. It is composed of cells which are migrating from the neural crest. Here is a neural crest. You already know that it is very potent tissue, giving rise to many organs, for example, sympathetic and another ganglia, and also mesenchyme of skull and teeth and cervical area. Pharyngeal arches are filled with this mesenchyme. So with very interesting tissue, neural crest, and in this area, in the area of genital ridge, it gives rise to a group of cell which is coming to the contact with the cords of coelomic epithelium and which is transforming into the medulla of the adrenal gland. So when I'll go to next picture, we can see cortex and medulla from chromaffin cells. Cortex is differentiating in two waves. It's very similar to the formation of gonads. There are also two waves, mainly in ovaries. There are two waves. There are primary and secondary sex cords, which are called also medullary and cortical. And here is something like this. So the first wave of the epithelial cords is forming acidophilic fetal cortex, which is very important for the secretion of steroid hormones during the fetal development, which are transforming to estrogens in the placenta. And then in the second wave, adult cortex is developing, which is replacing this original acidophilic, which is differentiating its three layers and which is secreting another steroid hormones and acidophilic fetal cortex, which disappears during early childhood. So that was the adrenal gland. I see that there aren't any questions. Yeah, okay. So I can go to next gland. Next gland will be thyroid gland. It is the biggest endocrine gland. Its slide has a number 76, and you can see that it's very, very different than all previously demonstrated glands. If you remember one of slides at the beginning of our today's stream, you probably know, or I'll show this slide once more, that this is the gland. This is the only gland where the epithelium is not trabecular but planar. And planar epithelium is lining surfaces. So this planar epithelium in this case is arranged in follicles. So it is the only follicular endocrine gland. And these follicles are visible in our slide. In the small magnification, we can see a lot of spherical structures, spherical sacs of variable size. Some of them are very small, some of them are very big, and all of them are lined with simple epithelium. This simple epithelium can be cuboidal, usually is cuboidal, but if this part of gland is very active, this epithelium can become columnar, and if this part of gland, if this follicle is less active or inactive, the epithelium is squamous. So it's every time simple epithelium, and its height can be very variable, from squamous through cuboidal to columnar. In the follicle, in the lumen of each follicle, is some eosinophilic material. It can be darker when the follicle is big, and it can be lighter when the follicle is small. And the name of this material inside is colloid. So there are follicles, thyroid follicles, and inside is a colloid. What is around? Around follicles is a reticular connective tissue. Again, the same situation as you can find in all other glands, endocrine glands. And when we look at it in high magnification, we will see many capillaries. Capillaries of thyroid gland are again fenestrated. Thyroid gland is one of the best vascularized organs, so we will see many fenestrated capillaries around each follicle. These follicles are closed spaces without any lumen, without any duct. They are separated from the surrounding connective tissue by continuous epithelial lining, and so the cells which are forming them have to secrete their products into blood capillaries, and that is why there are so many capillaries. You can see in this small magnification also capsule of a gland in this area. You can see that there is some adipose tissue, loose connective tissue, that there are vessels in the capsule, and this capsule penetrates to the interior of the gland, bringing vessels inside. So here is one septum with quite big vessels. So the thyroid gland is also quite well lobated. There are many lobes and lobules. Let's look at some of these follicles. I use higher magnification. And what do we see are really hollow balls, which are lined with, in this case, cuboidal cells arranged in one layer. But if you look at this bigger follicle upstairs, the cells become squamous. It is less active follicle probably, and there is sometimes possible to find follicles with higher epithelium, with columnar cells. No. Maybe here I see quite high epithelial cells where nuclei are ovoid and parallel with the axis of the cell. So let's say here columnar cells. And look at very many capillaries around. All here, these erythrocytes are arranged in capillaries. Some of them, no, some of them are artifacts here, but here is a lot of capillaries which are surrounding our follicles and which are supported by reticular fibers and reticular cells. So reticular connective tissue is forming the stroma. The cells which are forming our follicles are called follicular cells. So the main cells of the thyroid gland are follicular cells. So let's look at their function for a moment and also to their outer structure. Follicular cells are very interesting. They are able to produce proteins, they are able to secrete proteins, they are able to resorb the colloid which is inside. They resorb it by phagocytosis. They then digest colloid droplets by lysosomal digestion, and products of this lysosomal digestion are active hormones of the thyroid gland. That means T3 and T4, or triiodtyronine and thyroxine. So this cell, so let's take for example this cell, it can secrete proteins in this direction, it can engulf these proteins back to the interior, it can digest them and secrete small molecules, hormones to blood. So it has very many functional features, and I'll use a schema to elucidate it a little. It is in this presentation. I have to find thyroid gland here, and yes, I'm looking for this picture. So this is a picture showing functions of thyroid follicular cells. So this brick here is a follicular cell, and we can see two phases of its secretory cycle, and in the central picture is a resting or storage phase. So this cell can also sleep for some time and store products inside, and in this case it becomes lower squamous or cuboidal or squamous, and it is the case of this big hypoactive follicles. But in the left and right picture is a cell higher, let's say columnar, it is active cell, and in this left picture it is the active cell in the secretory phase or synthetic phase of the secretory cycle. The product of this secretory phase is called thyroglobulin. It is a protein, thyroglobulin. As it is a protein, it must be synthesized in rough endoplasmic reticulum, then it goes through Golgi complex, is situated in, they are writing here vacuoles, but I would rather say secretory granules, and these granules, or the content of these granules, is secreted to the colloid. Together with this, the cell is transporting iodide ions. These iodide ions are transported from blood through the iodine pumps, through the whole cell, then also to the colloid. But during this secretion to the colloid, iodide ions are oxidized into iodine, into molecular iodine, and thyroglobulin secreted into the colloid is then iodinated. So iodine is very important for this first phase of thyroglobulin synthesis. Then the cell can resting or not, it depends on the hormonal stimulation. These cells are stimulated by TSH from adenohypophysis, which we already described, and it can go to the second phase. If well stimulated, it can engulf particles of the colloid, like during phagocytosis. You can see how the cell is sending processes like pseudopodia and particles, droplets of the colloid are going inside. These colloid droplets then fuse with lysosomal transporting vesicles, and after this fusion, thyroglobulin, iodinated thyroglobulin, is digested. Protein is break down, and some of its amino acids, which are iodinated, can transform into T3 or T4, which is triiodthyronine and thyroxine, and they go to blood. They are transported by carrier proteins to all organs. They can go to the target cells through the membranes without any receptors, and they can digestly affect mitochondrial metabolism. So target organs of triiodthyronine and thyroxine are mitochondria of all organs, and they stimulate energetic metabolism. In this presentation, there is also second picture which is showing almost the same things. It's new in this presentation. I edited it there on Monday or Tuesday, I don't remember exactly. And in this left part, there are also steps of the follicular cell function: synthesis and secretion of thyroglobulin, uptake and transport and oxidation of iodination of thyroglobulin. Then dimers have to be formed because T3 and T4 are dimers of two tyrosines which are iodinated. Then lysosomal digestion and secretion. Sometimes there can be also alternative so-called transepithelial pathway of thyroglobulin transport without lysosomal digestion. So the follicular cells can secrete to blood not only T3 and T4, thyroxine and triiodthyronine, but also the whole protein, the whole thyroglobulin. So some thyroglobulin, some levels of thyroglobulin are bad. No, thyroglobulin. Some levels of thyroglobulin are found in normal blood as well, but mostly this transepithelial pathway is used during hypersecretion of or hyperfunction of the thyroid gland, so-called Bazedov disease. Okay. So that's about the function of the thyroid follicular cells, and there will be another electron micrograph which is in this presentation as well, so it's not necessary to skip to another presentation. I show it full presentation. So what do we see here? Is it possible to draw here? Yes, it is. So what do we see? is doesn't work, doesn't matter. F is a follicular cells. So let's say this is a follicular cell. It's quite small magnification of the electron microscopy, so we can see many follicular cells. Here's the third one, fourth one, and so on. Each of them has got visible nucleus, and the cytoplasm, and centrally is colloid. So this is colloid. You can see, for example, here or here down, that the follicular cells send microvilli to the colloid, so they increase their secretory or resorptive surface. Follicular cells are rich in organelles. We can see many mitochondria of normal crystal type, of course. There is rough endoplasmic reticulum and other necessary organelles for the synthesis of proteins, and there is also a droplet of colloid which is just fusing with lysosome, so it is the proof of lysosomal digestion of a colloid. And so then the product of this lysosomal digestion is going through the basement membrane to blood. For example, here is blood capillary. No, it is here written. So here is a direct contact of follicular cells and capillary and the pathway of the hormones. And look at this structure here. Here is suddenly something else. Here is a part of cell, and here is a small one. Two parts of a cell having many granules. Follicular cells have got some granules as well, because they need them for the production of thyroglobulin. But this cell here has got much more granules, so it is another type of cell. Its name can be C cell, it's also possible to find, or parafollicular cells. So there exists a parafollicular cell. So there aren't only follicular cells, which are the main cells of thyroid gland, but also parafollicular cells. What do we know about them? These cells can be included in the follicular epithelium, like in this case, but they can also be situated in the interstitial reticular tissue. By the way, here is a lot of reticular fibers. We can see collagen fibrils in this electron microscope. So they can be also here. Theoretically, we can find such cell in the stroma. So they are not every time included in the follicular epithelium. I'll skip to our slide, and I will try to show them. Follicular cells are a little bigger. Parafollicular cells are a little bigger and lighter than follicular cells. So I'll look for bigger and lighter cells in our specimen. They can be directly included in the epithelium, or they can be situated between. Here in this field, I see here maybe 2-3 cells. They can be in groups, 2-3 cells in the interstitium, or here I can see quite bigger and lighter cell directly in the epithelium, in the follicular epithelium. If this cell is in the epithelium, it never contacts the colloid. There is every time some process of neighboring follicular cell which is covering the apical surface of our C or parafollicular cell. So they are only they are situated only at the basal part of this epithelium, or they are situated in the interstitium. What do we know about them? They have many electron-dense granules. It was visible in the electron micrograph. They have got lighter cytoplasm, less basophilic. That means their protein production isn't so active. And both these features are typical for cells producing peptides. And really, these cells are producing peptides. A peptide of parafollicular cells is called calcitonin. And it is a hormone which is stimulating osteoblasts to build bone tissue, and during this synthesis of bone matrix, these osteocytes are using calcium ions and phosphate ions, and so they decrease the calcium level in blood. So the result of calcitonin function is formation of new bone and decrease of calcium and phosphates in blood. Here I see some question. How can we differentiate between follicular and parafollicular cells? As I said, parafollicular cells are a little lighter and bigger. They are not every time in groups. They can be isolated single cells. It isn't a rule that they must be in groups, but they are all the times lighter and bigger. So if you want me to find some another ones, I'll try. Wait a moment, I delete these drawings here. So let's look at it. For example, here is one bigger and lighter cell in the follicular epithelium, and it is parafollicular cell. Or there are not very many, so we can see follicles without them, and then suddenly we can see group. For example, here is the situation of parafollicular cell between follicles, and here is one in the follicular epithelium. Their recognition isn't easy, so just to know how to recognize them, it can be enough. So, thyroid gland is demonstrated according to my meaning, so we can go on. And we can go to parathyroid gland, slide number 77. Parathyroid gland is a small gland. You can see in this small magnification almost the whole organ. There are 4 parathyroid glands located in the dorsal capsule of the thyroid gland, so they are very, very intimately connected, thyroid and parathyroid glands. There are 2 superior to inferior parathyroid glands which are developing from another sources. I will show brief development in a moment. if you look at it in this small magnification, you can see again some capsule on the surface, which is common with thyroid glands, some adipose tissue around. Adipocytes are also inside. They are entering this gland together with connective tissue, septa and vessels. And the proper parenchyma of the parathyroid gland is again trabecular epithelium. So similar tissue as you can see in adrenal glands or in the adenohypophysis. This trabecular epithelium is forming such cords which are supported by reticular fibers, which we don't see in this magnification, and let's look at them in high magnification. Cells of parathyroid glands are very small, so we need the highest magnification to see them well. This is now highest magnification, and let's look at several cords of this gland. You can see very tight arrangement of cells, and the cells are very small. Sometimes parathyroid gland can resemble some lymphoid organ because the cells are so small that they can resemble lymphocytes. Their size is only 5, 6, 7 micrometers. That means the size which is similar to the size of erythrocyte. Here is, for example, an erythrocyte, and you can see that it isn't much smaller than the cells of parathyroid gland. These small cells of parathyroid gland, which are forming the most of cells of its course, are called chief cells. So these yellow cells are chief cells of parathyroid gland. They are the main secretory cells of the gland, and they are secreting special peptide which is called parathyroid hormone. Parathyroid hormone, abbreviation PTH, is a peptidic hormone which has opposite function than calcitonin. It is stimulating osteoclasts to resorb bone tissue, and so to increase the level of calcium and phosphate in blood. They also stimulate enterocytes in the intestine to absorb calcium salts. They also influence kidneys to absorb more phosphate, so they have complex function, but their main target cells are probably osteoclasts, and the resorption of bone is typical during their hyperfunction. So parathormone or parathyroid hormone is a hormone of chief cells, very small peptide secreting cells of parathyroid gland. If you look at this field carefully, you can see several bigger cells. I take red color because they are eosinophilic. They can be situated in groups, like in this case, or they can be individual. They are a little bigger than chief cells. They measure about 10 micrometers, but they have more condensed and smaller nuclei. So if you compare a nucleus of this cell and let's say this cell, it is one of chief cells. This nucleus is bigger, but the cell itself is smaller. These bigger eosinophilic cells are called oxyphil cells. Oxyphil cells. They aren't found in children. they develop and increase in number during life. At first they appear about at 8 years, then they increase in number, and it was thought for a long time that there are some degenerating cells, but when somebody looked at them in the electron microscope, brought a very surprising finding that they are full of mitochondria. So why degenerating cells are full of mitochondria? It doesn't make sense. So now we don't think that they are degenerating cells, but we don't know what do they do. We don't know anything about their function. We only know that they are increasing in number and that they are rich in mitochondria. So oxyphil cells are a little bit strange and mysterious cells of the parathyroid gland. There are just few cells, but few oxyphil cells, but we can find some areas of our specimen where the number is bigger, because it's probably a parathyroid gland from some very old person. So here is oxyphil cell, here is oxyphil cell, here is an oxyphil cell, here is another one, bigger eosinophilic cells, and the other ones are chief cells, which are secretory. Yeah. Okay. How thyroid and parathyroid glands are developing? Something was told about it in the chapter of digestive system, because they are developing together with pharynx. They are endodermal in origin. No, I don't want to change objective. I want to change the whole picture. I want embryological presentation, or maybe better is not presentation, but not presentation, sorry, but movie. Pharyngeal arches. You already know this movie. It was demonstrated in the digestive system. It was fortunately during this period when you were able to go to school, and so you have some knowledge about it. And now interesting things for us are sorry, I haven't got drawing. How to do it? Is it possible? No. So sorry, it is impossible to draw, so I just can show pharyngeal pouches. Pharyngeal pouches are important. You know that pharyngeal pouches are from endoderm. On the other side is the ectoderm, forming pharyngeal grooves or clefts or so, and in between is a mesenchyme forming pharyngeal arches with vessel, cartilage, nerve and muscle. But it isn't interesting for the formation of thyroid and parathyroid gland. Only pharyngeal pouches are interesting. So endoderm. There are 4 pharyngeal pouches. First, second, third, fourth. We are looking to the lumen of pharynx, primitive pharynx, and at the floor of the primitive pharynx there are several processes or tubercles which will give rise to tongue. Probably you remember that it is blue central is tuberculum impar. These two big here are the lateral lingual swellings. This small yellow is scopula, and this big yellow is eminentia hypobranchialis. It was already said during the formation of tongue. And let's look at this structure between tuberculum impar and copula. This is the place where thyroid gland will develop. So here will thyroid gland develop, and third and fourth pharyngeal pouches will be the places of parathyroid glands development. So let's remember this picture and let's go on. What happens? What happens in this area of pharynx? Yes, so pouch, and now we'll look at the fate of pharyngeal pouches. There are 4 pouches. There will be a longitudinal section of the primitive pharynx, so we will see the same picture as was before a while, and look at the events in the third and fourth pouch. They change, they increase, they divide. This third and fourth pouch divide into dorsal and ventral parts. And for us, more interesting are now dorsal parts. These 2 red and 2 blue dorsal parts will transform into the parathyroid glands. And what is interesting, these red parathyroid glands will be later inferior parathyroid glands, because they are then removed from the pharyngeal epithelium and they are transported deeper towards the thoracic aperture, and they are transported together with thymic primordium. This thing here down is thymus, and these 2 red structures here are inferior parathyroid glands, and they were transported together with thymus, and so they became inferior parathyroid glands. Superior parathyroid glands descended a little as well, but not so much, and they are from 4 pharyngeal pouches. So both or all 4 parathyroid glands are from endoderm of pharyngeal pouches. Superior are from the 4th pouches, and inferior are from the 3rd pouches. This pink moon-like structure here is a primordium of thyroid gland. I already said that it is developing from this spot between the tuberculum impar and copula, that means between first and second pharyngeal arch in the floor of pharynx. Next part of this video will show how does it happen, but now it's in the sagittal section. So here is this structure, thyroid diverticulum or thyroid primordium, which is developing at the place where we saw it before. That means between first and second pharyngeal arches in the ventral side of the pharynx. It forms diverticulum. This diverticulum is increasing, and we will see that it's elongating and growing down along the hyoid bone and then along larynx and trachea, and it reaches at the end a definitive position in the ventral side of the trachea, which was developing from the respiratory diverticle here. So now it is still connected, or long time connected with the pharynx by thyreoglossal duct. That means there is a duct connecting thyroid gland and tongue. Glossum is tongue. This thyreoglossal duct disappears, but something is remaining as a rudiment. It is pyramidal lobe of the thyroid gland, and foramen caecum on the surface of the tongue. And thyroid gland is now at the ventral side of the trachea. And now once more we'll see all together. So this is primordium, this moon is primordium of thyroid gland. These purple potatoes are primordia of thymus, thymic epithelium, and here are inferior parathyroid glands and superior parathyroid glands, and everything will migrate down. Everything will migrate down, and we can see these structures, these parathyroid glands on the dorsal side of the trachea, the thyroid, and there was one more structure, this red ball here, and you can see that it disappears, that it's dispersing into several particles and disappears, and it gives rise to parafollicular cells. Parafollicular cells of the thyroid gland are different in origin. They migrate to the tissue of thyroid gland during the development. They originate in the structure which is called, I'll show it here. Here it is written, which is called ultimopharyngeal or ultimobranchial body, and it is developing from the ventral, from this part of the fourth pharyngeal pouch, but the parafollicular cells are originating in neural crest, so probably cells of neural crest, this is again this our strange neural crest, are migrating from the dorsal side of the embryo to these ultimobranchial bodies, and together with these bodies they migrate to the tissue of thyroid gland and become parafollicular cells of the trachea. Sorry, of the thyroid gland. Okay, so I think it can be enough about development and also about parathyroid and thyroid glands. So I switch. How does it work? Yes, I switch this movie and we'll look at the last slide of this day. Last slide of this day is a slide which you already know, which you already saw. Probably you are recognizing what is it, because it is quite easy to recognize this gland, but I will help you. It is a pancreas. This pancreas was already demonstrated as the exocrine gland of digestive tract, and you know that there are Langerhans islets situated in pancreas, and now it's time to focus on the Langerhans islets briefly. This specimen is very rich in Langerhans islets. Probably your specimens you saw in the digestive tract didn't contain so many islets, so it's quite easy to find them here. There are, for example, this is an islet. This is an islet. Here. They are very variable in size. Some of them are bigger, some of them are smaller. They are dispersed in the exocrine tissue of the pancreas. Just for repetition, exocrine pancreas is the compound acinar gland having secretory acini. These dark basophilic structures, I use another color, are secretory exocrine acini, and they are all together connected with the system of ducts. So for example, one bigger duct is here, or here is a very big duct, but if we use high magnification we would see small ducts also everywhere here among these serous pancreatic acini. Serous pancreas or exocrine pancreas was already described, so we'll not go back to this. Let's focus only to Langerhans islets today. Langerhans islets, which I now increase, are composed of trabecular epithelium, so they have a structure a little bit similar to parathyroid glands or adenohypophysis. So here, or I use the highest magnification, it will be nice. Here is a perfect picture. You can see everything here. There are pancreatic serous acini now in high magnification. Even more we can see centroacinar cell. Here is a duct or another one. So that's everything, or here is one small duct. So that's everything belongs to exocrine pancreas, but it isn't interesting for us today. For us is interesting just this structure. And as you can see, it's composed of cords of epithelial cells. These epithelial cells are smaller and mostly lighter than the cells of exocrine pancreas. This is typical picture of cells secreting polypeptides. Pancreatic hormones are polypeptides. There are several polypeptides and each cell can secrete only one hormone, so there are also several types of secretory cells, but we don't recognize them in hematoxin eosin staining. So just small, slightly basophilic cells. Of course, everywhere around is reticular connective tissue, as usually, having vessels. So if we focus on the vessels, we can see here an erythrocyte, for example, vessels or capillaries of the endocrine pancreas are again fenestrated. Yeah, so there are fenestrated capillaries, cords of epithelial cells, so trabecular epithelium, and reticular connective tissue forming a stroma. These cells are not recognizable, but we know that there are at least 4 types, because there are 4 main hormones of the islet of Langerhans. We can look at the presentation where it's written. So, but not this one, it is development, but I switched it off, so wait a moment. Endocrine, it will be this presentation. This presentation, and at the end are islets. Yes, and what I wanted to show is for example this picture. Here it's very poor resolution, but it is a special staining of Langerhans islet with aldehyde fuchsin, as you can see here written, and this staining can help us with recognition. In this staining there are some red cells. Yeah. For example, here is a red cell, or here is a red cell, and it was recognized that the red cells stained by aldehyde fuchsin are sorry, why I do it blue? It was a mistake. Red cells have to be surrounded by a red line, and these red cells are A cells, or also alpha cells. We can see also blue cells. They are more numerous. Blue cells are B cells or beta cells. And there are also cells which don't stain specifically in this staining, and they are D cells and F cells, which are also called PP cells. So aldehyde fuchsin staining is one not very perfect method of their recognition. Another method how to recognize cells of pancreatic islets or Langerhans islets is immunohistochemistry. Immunohistochemistry can help us very much, because here we are using antibodies against hormones. We know that B cells, it is this case, B cells secrete very well known and the most important hormone of the Langerhans islets, and it's called insulin. They are the most numerous, and if we use antibody against insulin, we can see positive in this staining brown cells, which are forming about 70% of epithelial cells of the pancreatic islet. If we use another antibody, and it must be antibody against, oh no, it was a mistake, it must be antibody against glucagon. Another hormone of pancreatic islets is called glucagon. So if we use antibody against glucagon, we will see about 20% of positive cells. And we know that glucagon is secreted by A cells. So now we are visualizing A cells. Yeah, so B cells are the most numerous, A cells are less numerous. Together they form about 90% of cells, and the remaining 10% are, for example, D cells, which secrete somatostatin. So a hormone of D cells is somatostatin, and F or PP cells, they secrete pancreatic polypeptide. So immunohistochemistry is a perfect method how to recognize the cells at the level of the light microscopy. And we have another options. We have also electron microscopy, and electron microscopy can help us as well, because the islets of Langerhans are good for electron microscopy by the fact that the granules of their main cells are very different. For example, A cells having or secreting glucagon have got granules of this type. They have electron dense core, which is eccentric, and electrons moderately dense periphery. Cells secreting insulin, that means B cells, have got granules with these angular crystalloid structures inside. Cells secreting somatostatin, that means D cells, have got completely dense granules, and cells secreting pancreatic polypeptide, that are F or PP cells, have got typical dense core vesicles with central dense core and electron-lucent periphery. Here you can see also some minor cells found in the pancreatic islets, and who was studying enteroendocrine cells of the intestine and stomach maybe remembers that these cells are there as well. It isn't surprising, because pancreas is from the same embryonic origin as the intestine. It is from endoderm, and so the same cells belonging to DNES system can be in the intestine, stomach, and pancreatic islets. It isn't anything surprising. And when we look at the A real electron micrograph, it's too small magnification, you wouldn't see anything, but in this case you can see in this high magnification granules which have crystals inside. Look at it, this is a granule. Yeah, here is a nucleus of a cell. But here is a border, so here's another cell, and this granule has got crystal inside, and according to this finding we can recognize that it must be B. And next cell has got dense core granules, but these cores are not central but eccentric, and the matrix or peripheral zone isn't wide, it's moderately dense, and this is the proof that it is a cell having a glucagon inside. And the last picture of this area is a cell having completely dense granules, and in this case it's a proof, or almost proof, that it's D cell. And according to my meaning, it can be the end of our practical training of this day, the end of endocrine system. I see that there aren't any other questions, so I suppose that everything was clear. Thank you for attention. Thank you for your presence. Of course, many other students can look at it later because it will be saved in the YouTube. And I hope I will meet you, I virtually meet you tomorrow at 1 p.m. of the Czech time, when we'll try to write the test about the respiratory, urinary, reproductive system, development of urogenital system, development of heart and vessels, and apoptosis and cloning. There will be really just 10 questions. 10 questions. You already know that the result will be not taken to the credit. We'll just try if it is possible to write tests through Moodle. So come at tomorrow on Friday at 1 o'clock to your computers, log into Moodle, go to credit and examination tests, and wait for the email with password, and then you can write it. You will see your result, and we will write your result to the card, but as I said, it isn't a part of the credit. It's just training, and mostly for you it's training if you are able also without presence in the school to study, to study more topics together. So I recommend to fill the test without any open textbooks or Wikipedia or other sources. I recommend to work only with your memory and with knowledge you have in your heads. So see you tomorrow. Bye.